Hello everyone, welcome back to our Lee County Library Science Club. It's great to have you with us again. Just a reminder, please make sure you have your parents' permission and some supervision when you're doing these experiments with us. Last week we learned a little bit about cells, right? The small little building blocks that are inside every living thing that help our body function and make sure that its processes are running the way they're supposed to. Well, one part of the cell that we talked about was the nucleus. And we said something very special stays inside the nucleus. That is our DNA. Now, DNA is an acronym for deoxyribonucleic acid. Now, there's a lot of meaning in that name, but what's important for us to know, our DNA is the genetic material within your cells that tells your body how to grow and how to function. As we said, it's located inside the nucleus of almost every cell inside our bodies. But what actually is it? Well, it is what's known as a double helix, which if you took a ladder, if you had a big long ladder with the two sides and all the rungs on it, if you could somehow take both ends of that ladder and twist so that the ladder turned into kind of a spiral type shape, that is what is called a double helix. And each of the rungs on that ladder is what's known as a gene pair. Now, the sequence of the gene pairs, the types that are on each gene, those are the things that tell our body if we're going to be tall or if we're going to have, brown or blonde hair, what color our skin is going to be. All of these things come from the DNA. And there's also a partner to the DNA called RNA that actually tells each specific cell what its functions are supposed to be and how it's supposed to help the body to live and be healthy and to thrive. But today, we're going to be paying attention to the DNA. Now, as you can see, I have a lot of stuff in front of me here, so we're not really going to talk anymore about DNA because it's a bit of an involved experiment, so we're just going to get right into it. Now, what are we going to need? You need some isopropyl alcohol, salt, dish soap, water, a strawberry, a Ziploc bag, a strainer, a larger container like a bowl, and then a small skinny container like this glass. You'll need some measuring spoons and some tweezers. Now, like I said, that's a lot, so we're going to take it kind of slow and make sure we understand each step as we go along. But by the end of this, you are actually going to be able to see the DNA of this strawberry. Sounds pretty exciting, huh? Let's get to it. Now, the first thing you actually have to do is take your isopropyl alcohol bottle and put it in your freezer. Don't worry, we'll come back to it later, but the alcohol being chilled will really make sure this experiment works properly. Now, I have pre-measured my water, but you're going to want to make sure you have 6 tablespoons of water in your cup. And the first thing we're going to do is use this water to prepare what's known as our extraction fluid. So the extraction fluid is going to be a combination of our water, soap, and salt, which we will use to pull the cell materials out of the strawberry. Like I said, we're going to start with the water. The water is our solute or our medium for the reaction. Next, you're going to take your dish soap and pour 2 teaspoons of dish soap into the water. Next, we're going to take our salt and you're going to do a 1/4 teaspoon of salt into the water and soap. So what we're doing with these, like I said, the water is the base for our extraction fluid, but our other parts have important functions. You want to stir them all together. The soap, because of the way, we've talked about before, soap molecules are kind of special. And because of the way they're made, the soap can actually dissolve the cell membranes. So inside the strawberry, just like inside us, are lots of cells. And they all have the cell membrane with all the other stuff inside. So the soap will dissolve that cell membrane so that the stuff inside the cell can come out. And then the salt does a similar function on the structure of the DNA. It actually denatures the proteins that hold the nucleic acids together. So the salt and the soap together will help to separate the DNA from the rest of the cell. Next, we need to take our Ziploc bag. You're going to put your strawberry into your Ziploc bag, and you're going to pour the extraction fluid onto the strawberry. Next, you want to make sure there's really no air left in that bag. So now you've got a strawberry in the extraction fluid in your bag. And here's the fun part. We're going to smash that strawberry until it's nothing but mush. Ready? Now while you're doing this, if the fluid kind of gets all over the bag, that's okay. You want to make sure you get your strawberry mashed as finely as possible, and then make sure that it mixes well with the extraction fluid. So you can see right now there's still some big chunks in there. So I'm going to keep mashing until all those big chunks are gone. Now if this process takes you a while, that's okay. Take your time and make sure you really get it mashed so that you can definitely have some strawberry cell material to work with. So once you've mashed it up and there's not really a lot of big chunks left, this is where we need our strainer and our bowl. You need to strain your extract through the strainer into the bowl so that only the liquid gets in your bowl. Now this part can get a little messy, so just try to be careful. So as you can see, I've still got some mashed strawberry here. So I'm going to take my spoon and really press it through there to get any more of the liquid into my bowl. Now once you've really made sure you got all the liquid out of there, there's a little bit too much surface area in our bowl here to be able to pull the DNA to the top. So we're going to take our other cup that we had and gently pour it in there. And now we need to go get our isopropyl alcohol out of the freezer. Next, you're going to take that isopropyl alcohol, pour in 2 teaspoons of the isopropyl alcohol. And what this does, the DNA is not soluble in the isopropyl alcohol, whereas the rest of the cell material is. So it's actually going to force the DNA to separate from the rest of the stuff that's in our cup. So you can see floating along the top there some nice white strands. So looking at it from above, you see all those white strands floating on the surface? Those are actually our DNA strands. Now I was perhaps a little bit too aggressive when I was pouring the alcohol in, and I think I broke them up a little bit too much. If you're gentle about pouring, you'll actually be able to pick some of these pieces up with the tweezers. Now I did have some trouble with this the first few times I tried, and I learned that you need to be pretty specific with your ingredients. So the salt that you use needs to be regular non-iodized salt. A lot of the table salt that we eat has added iodide to it. We need to get the salt without that. Your isopropyl alcohol needs to be a high concentration. So this 70% that I have only worked mildly well. If you have the 90 or higher percent isopropyl alcohol, that will work much better in getting clear, defined strands that you can pull out of the cup. Now, a process similar to this can actually be used on all kinds of different cells. Different fruits, vegetables, plants, even animal cells. This type of thing can be used to extract the DNA from all different kinds of cells. But for us, strawberries are much easier. It precipitates at a much higher rate, and also strawberries are one of the few organisms that has 8 pairs of DNA inside each cell, rather than us, which have just 2. Now, it's not hard to realize how important this type of research could be, right? We know so little about how our bodies actually learn and grow and develop, and studying DNA and actually being able to look at the specific genes that are on a strand is an incredible way to learn about our human bodies and just about the way life and organisms work in general. I hope you had a lot of fun with this, and we'll see you next time. Thanks for watching. Bye!